Hey there, and welcome. Today, we're digging into something pretty amazing, the hidden engineering that acts like a guardian angel for our DNA.
So just stop and think about this for a second. Every single time one of your cells divides, your entire genome, we're talking all 3 billion letters of it, has to be copied perfectly. Over your lifetime, that happens trillions of times. It's just a mind-boggling number. So the big question is, how does this incredibly complex instruction manual not just fall apart and turn into complete gibberish?
Now you'd think the answer would be in our genes, right? You know, the parts of our DNA that actually code for proteins and do all the stuff we learn about in school. But nope, that's not it. The real solution is hiding in plain sight in what scientists used to dismiss as junk DNA. Turns out, these vast, super repetitive sequences are the real unsung heroes of the genome.
So, there are actually a few different kinds of these repeated bits of code. But today, we're going to zero in on two of the absolute superstars: telomeres, think of them as protective caps on the ends of our chromosomes, and centromeres, which are like the anchors that make sure our genetic material gets sorted correctly. These two, they're basically the genome's guardian angels.
All right, first up, let's talk about the frame code problem. This is a huge challenge in biology, this idea that our DNA actually gets a little bit shorter every single time it gets copied. It even has a name, the end replication problem.
Okay, so picture this. The machinery that copies our DNA, it's kind of like a zipper, but it can't quite zip all the way to the very, very end. So with every single copy, a tiny little piece gets lost. It just falls off. And if there wasn't a solution for this, that relentless shortening would eventually start chewing into our actual crucial genes. And that would be absolutely catastrophic.
And this is where our first hero, the telomere, swoops in to save the day. It's basically just a long, long stretch of repeating code that forms a protective cap at the end. You can think of it as disposable DNA. It's meant to be worn down so that our important genes aren't. And what's really fascinating is that this shortening process also works as a kind of molecular clock for the cell. It's directly tied to aging. And when this system goes wrong, it's involved in some really serious stuff, like cancer. So yeah, this is pretty fundamental to our health.
But wait, the story gets even cooler. Our cells actually have a machine to fight back against this shortening. It's an amazing enzyme called telomerase, and what it does is it carries its own little blueprint, an RNA template, and it uses that to add those repetitive DNA sequences right back onto the end of the chromosome. It's literally like a tiny construction crew that's constantly rebuilding that protective cap, making sure it doesn't get too short.
So what would happen if this little construction crew just stopped working? Well, scientists did exactly that experiment. They engineered some plants, Arabidopsis plants, to not have telomerase. And the results? Well, they were devastating. For the first few generations, everything looks more or less okay. But then, around the 6th or 7th generation, you start to see this widespread degradation. By the 9th generation, the plants are sterile and they're tiny. And by the 10th, it's lethal. Game over.
And wow, this picture just says it all, doesn't it? On the left, you've got a perfectly healthy, normal plant. And on the right, that's the plant from the 9th generation, the one without that telomerase enzyme. The difference is just it's night and day. It's such a powerful visual reminder of how absolutely critical these little protective caps are for, well, for life itself.
Okay, so we've got the fraying ends problem sorted out. But that's not the only massive logistical headache our cells have to deal with when they divide. Now we've got the chromosome sorting challenge. So here's the deal. Before a cell can divide, it has to double its entire DNA library, creating 2 identical copies of every single chromosome. Then, it has to perfectly pull those copies apart so that each new daughter cell gets one complete set. So how on earth does it manage that incredible feat of organization? How does it do this without losing anything or creating a complete tangled mess? And the answer brings us to our second guardian angel, the centromere. The easiest way to think about it is like a handle that's built right into the chromosome. It's this super dense, compacted region where a big protein machine, it's called the kinetochore, can grab on. And that machine then becomes the anchor point for all the molecular rotes that physically pull the duplicated chromosomes to opposite sides of the cell.
And when I say it's a region, I'm not kidding. Just look at the sheer complexity here. What you're seeing is a map of the centromere from just a single chromosome in rice. This isn't just some tiny point on the DNA, it's a vast landscape. We're talking nearly 2 million DNA letters, built from thousands and thousands of repeating sequences. It is an absolute masterpiece of structural engineering.
And that, right there, brings us to a huge scientific challenge.
Imagine trying to solve a jigsaw puzzle where almost all the pieces are the exact same shade of blue. That's what it's like for scientists trying to sequence a centromere. All those repeating blocks are almost identical. And for decades, these regions were just blank spots on our genome maps. We knew they were there, but we had no idea what they looked like.
So how in the world did we finally get a detailed look at these incredibly complex hidden structures? Well, it took a massive, coordinated effort. And that brings us to the final part of our story today, mapping the blueprint with the Rice Genome Annotation Project. So the Rice Genome Annotation Project, or RGAP for short, was this huge international undertaking. The goal was to create the very first high-quality, fully annotated, and that's the key word, annotated, reference genome for rice. And why rice? Well, it's one of the most important food crops on the planet. And annotating meant they weren't just going to read the DNA letters, they were going to identify every single gene, every repeat, every hidden feature, the whole shebang.
And this project really became a cornerstone for plant biology. It was a massive international collaboration, especially with researchers in Japan. And together, they created this single, unified reference genome that everyone could use. It's now based at the University of Georgia, and it's still a go-to, absolutely critical resource for scientists all over the world who are studying not just rice, but all kinds of different plants.
And just to give you a sense of the sheer scale of this work, look at that number, 39,049. That's how many different gene locations they identified in just one data release. And that doesn't even count all the mobile bits of DNA called jumping genes. This is just meticulous, painstaking, large-scale biological accounting. It's incredible.
And what's so great is that this work brings our whole story full circle. Because of these incredibly detailed maps from RGAP, researchers could finally go in and characterize the telomeres on every single rice chromosome. They could measure their exact length, study their structure. It took the idea of a telomere from this abstract concept and turned it into concrete, hard data.
And yep, you guessed it. They cracked the centromere problem too. The project completely sequenced the centromeres from two different rice chromosomes, number 4 and number 8. They did something that was, as this quote says, notoriously difficult. They filled in some of the biggest, baddest blank spots on the genomic map, and in doing so, they revealed this hidden architecture that's essential for life to even work.
So, in the end, projects like RGAP have totally transformed how we see the genome. They've shown us that the stuff we used to casually dismiss as junk DNA is anything but. It's absolutely essential for survival. Telomeres and centromeres, they're just two amazing examples from this hidden world. And it really makes you wonder, doesn't it? What other vital, incredible secrets are still hiding out there in the vast dark matter of our DNA, just waiting to be discovered?